turn the recorder on. Okay, so these are important because no matter what, the antibiotics that you're going to learn, they're going to fall into these categories. And then I have those listed out, and this is a mess. Then I want you to know big picture stuff like what is drug resistance? Where does it come from? Why do we care? Why is it a big deal? And that's kind of like where does resistance come from? So that's in there. Oh shoot. Now the stuff about I have all the different drugs listed on here.
Yes.
And I don't expect you to memorize all of them. What I want you to do, and I guess I can make sure that is clear, is you are going to look at the ones on that sheet I gave you. So there's a page.
Yes, so just memorize that sheet.
Yeah, because there's so many of them, you could spend a lot of time memorizing them. But I made this little summary sheet to try to make it straightforward.
8 of them, okay.
So this is what I'm more worried about you guys knowing, because I think this will apply maybe when you go other places, hopefully.
Okay.
So these are the big ones. So I need to make sure that that's clear because it's not necessarily clear with the PowerPoint notes. But these are the ones that I want you to understand how they work. And it's pretty much an example of each type.
Okay. Yeah.
So so you don't have to memorize the ones in the PowerPoint and all those details.
Okay, I will do that. I haven't started studying that chapter yet. I mean, I did when
it's hard to
working on it, but this is probably gonna be left at the end because it's a lot.
To me, just memorizing them, I think is hard, and I don't know that it's super duper useful.
Okay. Yes.
Because if you're going to nursing school, you're going to do one thing. If you're doing dental hygiene, you're going to do another thing.
Yeah.
If you're a biology major, you're doing something else. So I'm just trying to hit the big things that are in here because they're going to teach you more about it as you get into your specific field. So I just want you to get the big picture kind of stuff.
Gotcha.
Yeah. So do you have specific questions? Kirsten usually has something specific.
Let me see. I'm looking to.
I do have a question, and I think I might have messed up in my notes, but I have bacitracin down as a cell wall inhibitor, but I think in the notes that you gave us, it says that it's a cell membrane inhibitor.
Oh, well, did you look in the book? What does it say? Let me see what it says. I'm looking in the book to double check. Bacitracin. Oh, I'm in the wrong chapter. Oh, shoot. Okay. Or we can Google it. Let's Google it. See what they say.
It says, okay.
Never mind. What does it say?
In the book, it says that it disrupts cell membrane. So I think I just have it wrong in my notes because I have it down in my notes as bacitracin, but then in the book, it's down as polymyxin.
Polymyxin. So yeah, polymyxin, they do membranes.
Okay.
But does bacitracin, let's see. Huh. All right, let me look. Bacitracin. polymyxin mode of action, mechanism of action. There we go. So it says it inhibits bacterial cell walls. Huh.
So are bacitracin and
Polymyxin.
not the same thing?
Polymyxin is one thing, and bacitracin is polymyxin and something, and like neosporin.
Oh.
Yeah, bacitracin is also formulated as an ointment with neomycin and polymyxin B. So bacitracin is more than one thing. It's a mixture. So polymyxin is cell wall. So they might have added something to it to make it bacitracin is a combination of 9 bacitracins. A bacillus, it came from a bacillus. But then down here, topical formulations formulated as an ointment with neomycin and polymyxin B. I thought it was mixed with more than one thing. Kind of like neosporin is more than, I think neosporin is more than one thing too.
Okay.
Yeah, I don't know that. I try to ask more big picture stuff than little tiny details.
Yeah.
But yeah, right. I I don't like memorizing these for some reason. They don't stick in my head. The only ones that do are penicillin and cephalosporins and sulfa sticks, the sulfonamides. The rest of them fall out repeatedly.
For
What else? What
for
else?
the exam 2 coming up, what chapters is it on?
I know, that's a good question. I was just looking at it. So on the syllabus, it says 8 and 9, right? Which would be the bacterial genetics. And then I have 11, which is viruses, sterilization and disinfection, and antimicrobial therapy. So really, I guess that's where I should stop. The next stuff is host microbe, and that goes more with epidemiology. So I guess I'll just do it on those like 4 chapters.
The one that's posted for this week is going to be a next exam.
I'll put it on the next exam. Will
Okay.
that mess you guys up?
No,
No.
that's good.
Okay.
I think that's better.
Okay.
Can we talk a little more about quorum sensing?
Yeah, quorum sensing. Wait, hold on a minute. Okay, so quorum sensing. Who talked about, did we talk about it last week? I talked about it with somebody.
Yeah, I talked about it with you last week.
Okay. So
I can listen then. If you...
No, let's talk about it because
It won't hurt to talk about it again.
it won't hurt to talk about it at all. Okay, so quorum sensing. So a quorum is the idea, it's a certain number of something. So like I think Congress has a certain number of people, of Congress people that have to be there in order for a vote to count. So like you have to have a 2/3 majority to vote in order for something to actually stand. Okay, and that's called a quorum. So it's just like it's a percentage of a group that has to be there. So some bacteria have this thing, and they started calling it quorum sensing. Okay, so what the idea is that some organisms, you have to have a certain number of them present in order for them to start making a toxin. So what they're trying to work on, and this is like a relatively new idea, maybe like 5 to 10 years old, maybe not even that old, maybe probably 5 to 10 years old. So the bacteria, what they have to do is what they do is they'll communicate with each other. So if this is bacteria, they'll produce molecules, and the molecules have to go and they have to bind to a receptor on another bacterium. And that's how they detect how many are actually present and in the environment. So once they hit a certain point and they have a certain amount of the orange stuff, which is a chemical, once they have a certain amount being produced, then they'll start to make the toxin. So then they'll start releasing a toxin that can actually cause disease to occur. Okay, and they figured a whole bunch of this stuff out. So they figured out like what molecules it is and what the receptors are and what genes control whether the molecule is produced and stuff like that. But what they found is you have to have so many of these like orange molecules present in order for them to start making the toxin. So they're looking at drugs that would either stop the formation of this molecule or it would block it binding to another molecule or whatever. Somehow it's interfering with the idea that the bacteria can tell how many are there. Does that make any sense?
Yeah.
So it'd be like,
That's
like
density, right? All those molecules.
yeah, yeah. So what it does is it doesn't kill the organisms, but it keeps them from talking to each other.
Okay.
So then they won't make the toxin if they don't know that the other ones are there.
And if it's not enough of them, they don't produce anything.
Yep. They don't make the toxin. Yep.
Okay.
So like one of the really bad E. coli's. So the E. coli, the one that it was found, so it's it's O157H7 is the really bad one that's found like in feces. So if it's found in like, sometimes it's like it started out originally with hamburger, right? So if you ate hamburger that was undercooked, this organism, if it reaches a certain number, then it produces a toxin and it causes bloody diarrhea and can cause kidney failure. So they figured out that if they can stop this kind of E. coli, this strain of E. coli from communicating with each other, then they don't make the toxin that causes the bloody diarrhea. So you don't necessarily have to kill the organism, but you have to stop it from making what the thing that makes people sick.
Okay. Okay.
So it's kind of new. So like if you go Google, let's see, like I had this student, she went, and I already told Kirsten about it. She went to work at NIH as an intern, and let's see, and she went to a seminar and they were talking about quorum sensing, and she's like, why didn't I learn about this? I should have learned about this in biology or whatever. So here's a paper from 2010, so 10 years ago, cell signaling in Pseudomonas aeruginosa. So Pseudomonas is a really bad, can be a bad human pathogen. It depends. Like it's usually for people that are immune compromised, but if they can stop it from talking to each other, then it doesn't produce the toxin. So it's not as bad. So it's just a different way. Instead of using an antibiotic that might develop resistance, if they can stop it from talking to each other, it's a way to control it without having to use antibiotics. So they use it for cholera.
Is it widely used? This.
No, it's really new. It's really,
Oh,
really
that's
new.
why antibiotics still dominating. Okay.
Yeah, it's really, really new. So let's see. She was quorum. Oh, look here. Let's see what this. Somebody's got a picture. Yeah. See, so somebody's figured out. Like we don't have to go over that. All like this. I had a student talk to my class, and one of the biotech classes, and he was working on the signaling and how they actually are communicating and blocking it or detecting it. So one of the things he was looking at was diseases that cause sepsis. So when the bacteria are growing in the blood and somebody's getting ready to be septic, which being septic, if the bacteria are multiplying in your blood, it's not good. It's really, really bad. And he was trying to develop a way to figure out before someone is septic if the molecules, like the things that I drew that were orange, if he could detect those. Does that make any sense at all?
Yeah. Yes.
So it's just different way. It's just a totally different way of trying to look at,
All bacteria. Okay.
yeah, what's going on. Although these aren't. Yeah, this stuff is not. This is like over.
Too many details. Yeah.
Yeah, we don't need to know all those details, but I'd love to find it like an easy picture. I'll have to keep looking. Well, what's this one? This is like trying to get it to respond. Yeah, never mind. Don't worry about it. That. But it's cool because they're running out of antibiotics, right? So there aren't many antibiotics left. They can't find new ones. There haven't been any discovered in a really long time. So they're trying to come up with different ways to kill bacteria or to protect people from bacteria or the toxins that they make.
Okay.
Because we're just running out and there's antibiotic resistance to everything everywhere. So they're just trying to come up with different ways to try to kill the organisms.
Okay. Thank you. That was helpful.
Yeah.
On that, because I'm just looking at the like the study guide that you posted
Yeah.
at the beginning of each, and with the drug resistant, one of the things is how can we limit drug resistance? Can you go over that?
Yeah. So how to limit it?
How to limit? Don't take it.
The ways that we can limit it is to, well, is there a chart with five? Like there's like 5 ways. Well, maybe not. Different ways that they try to limit it is one is not use them unless it's absolutely necessary.
I thought there was a chart. There was a.
Yeah, I thought I had a chart too. I'm looking. Maybe
This
I'm.
was one of the questions I think on.
Yeah, I put it on the worksheet probably.
Yeah. Yeah, that was one of the.
So not use antibiotics unless you know they have a bacterial infection. Because it won't work for viruses, right?
Yeah.
So people go in, and I'm guilty of it too, right? I feel bad, and I just want the doctor to fix me. And I'll go in there, and I'll be like, oh, or I would do it with my kids. Like, oh, I think they have this. Can we get antibiotic? And they're like, well, we've just been seeing a virus around. So if you take an antibiotic and it's not necessary, then you're taking the bacteria that are already around and you're exposing them to that antibiotic. So then the next time when you need the antibiotic, it might not work, right? Because the organisms have already seen it. So that's one reason. So the other thing is to complete the course of the antibiotic. So I don't know that that's spelled right. So if they gave you 10 days and they say take it 2 times a day for 10 days, finish it. If you don't finish it, what can happen is you kill off the bacteria, right? So if these are the bacteria and you stop taking it after 5 days, you've killed off some of them, but you haven't killed off all of them. So then the ones that are left will have seen the antibiotic, and then they might be the ones that are resistant to it. So you have to take it for the whole dose, and everybody stops after 7 days. You stop after you feel better, and and then you end up with there's a little bit left in there that haven't been killed, and they are resistant to the antibiotic because they didn't die right off the bat. And maybe another day or another couple doses would have killed the rest of them off or reduced them so that your body could get rid of them. But everybody stops early, and then these guys end up being resistant. So that can be a problem. I know, I'm trying to think. I can't find it in the book.
I have that answer. It says, describe 2 antibiotic synergetic examples, streptomycin, penicillin. Would that be helpful?
Which one?
I just answered this question. This was question 11 of the
Okay.
worksheet that you gave us. Not worksheet, the questions to help.
Yeah.
I have, we can limit drug resistance by controlling environment, high dose. The 2 that you already told us about, and then it says 2 antibiotics together.
Oh yeah, that's it. So the synergistic effect is what they'll do now with some things that are antibiotic resistant. So they'll give you 2 antibiotics at the same time, like you said, penicillin and streptomycin, or 2 that they add together.
How would that limit that resistance if you take it? You're still taking it.
But you're not. What you're doing is you're attacking it 2 ways. So what it ends up happening is you're more successful at killing more of them.
Okay.
So then, like, this will inhibit the cell wall synthesis, right? And the streptomycin is going to go in, yeah, and interfere with protein synthesis. So by getting this one in, by breaking the cell walls and doing this, it's more effective, and you can kill more bacteria.
Okay.
So that's one of the big things that they're doing now is they give kind of like a cocktail of antibiotics rather than just one.
What's the strongest antibiotic available?
I don't know, because there's always something new coming out. There's some that we can Google it. Let's see what they say. What Google says.
I wonder if the doxycycline probably.
Yeah, right. What's scary is there were in different countries they use them differently, right?
Yeah.
And so there were some antibiotics that were relatively new, and like the United States, and I don't know if I talk about this in the lecture or not, the United States reserves some antibiotics for use only in people and not in animals. And like in China, they have pretty much used every antibiotic on farm animals and they've developed resistance to everything. So there aren't any left in China, the last I read. So,
Wow.
yeah, right. Strongest, let's see what they say. What strongest antibiotic for staph infection? Like, let's just put broad spectrum.
Well, maybe it depends what you're treating.
Yeah, let's just put broad spectrum though, because that would be, so examples of broad spectrum, but that's just broad. It's not.
Yeah, depending on what you.
Yeah, it depends on what you want. Huh. Cipro. This is saying Cipro. when this is the best use of new broad spectrum antibiotic from 1990, it was Cipro. So Cipro is a fluoroquinolone, and it is the one when the anthrax stuff was going down. That was the one they were saying. So these fluoroquinolones are really strong.
Yeah.
So hold on. Cipro is the one they were telling people to kind of hoard when the anthrax stuff. So the stuff from when the World Trade Center was being bombed, and they thought there was there was bioterrorism going on because people were sending anthrax spores. So Cipro, it's a fluoroquinolone, and it was used probably too broadly. Like they just kind of were like, yeah, have Cipro. Here, take Cipro. It'll kill everything, right? And it does kill everything. But now they know that it actually causes connective tissue damage. So there are people, like I've even had some students they're not very old, but they had Lyme disease, and they gave them Cipro for it, and it damaged their tendons and ligaments. So they have like long-term problems because of these. They were on Cipro for a long time. So it depends, right?
Yeah. Okay, depends on the target.
Yeah, it totally depends. And some things, right, they only will give to you, it has to go in through an IV and you have to be in the hospital while they do it because they don't want it to affect your kidneys because it's so strong.
Okay.
Yeah, what else?
Can you explain how antivirals work?
Yeah. So it depends too. So some antivirals, it depends on what kind of virus it is, how they work. Some are kind of like anti-metabolites. But what they do is they might look like a nucleotide. Okay, so they might look like an A, T, C, or G. Okay, but there will be something different about it. Okay, so it won't just be an guanine or a cytosine. It'll be a guanine or a cytosine that's got something different on it. So when the virus goes to do replication or transcription, it'll stop the production of making the DNA or the RNA. Does that kind of make sense? So where it should be picking up, so if you have A, T, C, C, G, A, T, T, right? And you're supposed to be going along and it's just supposed to be being copied, right? If we are treating it with something that has a C and the C is altered, okay, it'll stop this process and then we won't be able to copy it anymore. Okay, so it totally like stops that. So they call them analogs. They call them analogs, like purine or pyrimidine. So they look like a nucleotide. Okay, now some of them, it depends on what the virus is. Okay, so some of them might be able to, so if it'd be cool if they could come up with, okay, so this new coronavirus, right? Some of them will stop, if these are your cells, say these are the cells in your lungs. So these are the cells in your alveoli. Okay, and this thing's got to come up and it binds to a certain receptor here. And this actually, it binds to these things called ACE receptors. Okay, so if it binds here, if there's something that we could do to block this from binding to it, then it can't get in. So they try all sorts of different things. So in order for the virus to get in, right, it has to absorb, or it has to attach, it has to absorb, like all these different things have to happen, like the 5 different things have to happen. So they make antivirals to stop all of the 5 things. So some of them are more effective than other things. So like they're making monoclonal antibodies. So they're making antibodies that might attach to this, and then it won't be able to get into the cells. So if you can't get into the cells, you can't cause disease in the cells. So that's another example. There's also things, so HIV is a retrovirus, right? So what it does is it starts out as messenger RNA, and then it has to be made into DNA. So it has an enzyme called reverse transcriptase. So they've actually been able to find a drug that stops this. And then you can't make DNA from mRNA. Another thing is interferon. And that's considered an antiviral too. And interferon is something that we naturally make in our body. So our white blood cells produce it when we're exposed to a virus. So what they've come up with is they've come up with a way to actually synthesize interferon, and then we can give it to people. So there's all sorts of different ways. Sometimes they're hard on the body. So like with HIV, they use a cocktail of drugs, and the cocktail involves things like this, right? Things that might bind to the receptors on HIV and things that do this. So it's not hurting the person taking it as much as it would be if they just gave them one drug.
Okay.
Yeah, sorry, it's not like a real straight answer. There's lots of things going on. What else?
What about antifungals?
Oh, antifungals. I don't have, you know what, I don't even think I asked you guys any questions about them. And I probably should have, but I didn't. I know. I figure all the antibiotics, there aren't that many fungi that actually cause problems in humans.
Okay.
One thing that's hard with antifungals is because they're a eukaryotic cell, like we're eukaryotes, that it makes it hard to control them without having side effects. I don't even know that I have any questions on it, on the tests. I don't ask you anything about it.
Could you go over, and this is from the sterilization chapter, but I'm just looking at it for the test. Can you go over what the different chemical disinfectants do? And there's, I think, 9 or 10 of them.
Yeah. So like soaps and detergents and surfactants and acids. Is that what
Yes.
you're thinking? Okay. So, and there's a table, right, in the book.
I think so, yes.
Yeah. Okay. So soaps and detergents are the same thing. Okay. So those are considered surfactants. And what they're going to do is they're going to, they disrupt surface tension, and they're going to damage the cell membranes, the plasma membranes. So damage plasma membranes. Okay, so now when they talk about the coronavirus, right, the coronavirus is enveloped. So it has this membrane around it. So if you wash your hands with soap and water and, you do it for 20 seconds, it's going to disrupt that membrane, and that's going to destroy the virus. Okay, now on here, they have, okay, this chart, they have surfactants and soaps and detergents as two separate things. But surfactants, I consider them kind of the same thing. So a surfactant is what makes a soap a soap. So what they are is they're charged molecules, and they do pretty much the same thing. Okay, now if you look at acid, right, if something is an acid, what it's going to do when it's going to lower the pH, right? When you lower the pH of something, it denatures proteins. Okay, so it breaks it apart. So you lose that three-dimensional structure. Okay, so then bases or alkalis or bases, they're going to do the same thing, except they're going to raise the pH. Okay, and soap is made out of like lye, right? And some things are just really, really basic. So if it's really basic, it does the same thing. It denatures the proteins, it breaks them apart. Okay, now heavy metals is also on here. So heavy metals were actually, they were some of the earliest like disinfectants and sterilants. So heavy metals, what they do is they're gonna, essentially they break down the proteins. But think of things like silver, copper. I mean, they have selenium on here. But they're going to destroy the three-dimensional structure of the protein. So the proteins don't work anymore. Enzymes don't work anymore. Does that help? Is that making sense?
Yeah.
Is this what you want?
Yeah, I think because in my notes, I just have an example of them, but I don't have what they do.
Okay. All right, cool. All right, so now heavy metals, that's what they do. Then we have halogens. So remember, halogens are things like chlorine, iodine,
Right.
yeah, bromine, right? Bromine. Okay, so what these do is they're going to oxidize.
Cell component.
Yeah, cells, right? So when you add too much oxygen to it, or so what we're doing, not adding oxygen, but what we're doing is we're changing the amount of electrons there. So when we do that, when we oxidize it, so they're losing electrons. So they're going to lose electrons. So they're not going to function, right? It's going to destroy the structure of the molecules inside of it. Okay, so things like that. So chlorine, so the big thing on halogens, one of them is sodium hypochlorite. which this is what bleach is. Okay, think about the pool and chlorine, a hot tub and bromine. Iodine is they use on wounds. So they'd put it on a boo-boo and it would look orange. That was before like Neosporin was available. If you go to have surgery and they give you, oh shoot, what's the stuff they have you rub on it? It's bromine. Oh shoot.
I'm pretty sure it's iodine.
Yeah, it's iodine. It's betadine, right?
Look at that.
Betadine. So they give you now they give it to you in like a little wipe. Okay, so that's gonna oxidize the cells, the bacteria, and destroy the protein. So none of the molecules will work anymore. Okay, then alcohol is a big one, especially right now, right? Because of all the hand sanitizers. So alcohol, if it's mixed with water, it denatures proteins.
Mm.
It breaks them apart. Phenols are nasty, but they're around. Okay, so phenol is going to break apart the membranes. Okay, so, and then it's just bad all around, right? It denatures proteins. Okay. I think Lysol has phenol in it. Like the book lists some amyl phenol or a hexadine gluconate.
Cresol.
Okay, now oxidizing agents, that's like hydrogen peroxide. Okay, so the peroxide, what it does is it'll denature proteins, and the way it does it is it disrupts disulfide bonds. So that means we're going to denature proteins again. So mainly we're breaking down proteins or we're breaking down cell walls. Okay, now alkylating agents. Alkylating agents. So examples of those, some of those are really bad. Like formaldehyde is an example. Formaldehyde. Ethylene oxide. These are really, really bad. You don't want to use these unless you have like the appropriate PPE, like you have on a mask that filters it out. But what they're going to do is they're going to break down proteins, so denature proteins. And DNA and RNA. So they're nasty. They're really, really nasty. Now, okay, so another thing, and the last one on here is dyes, okay? And these are things like crystal violet, and they use this other one, acridine orange. And what it actually does, if it can interrupt cell division. And if it's binding to the cell wall, I can't spell, guys, division. Okay, and cell wall formation. they use that in animals. So sometimes like in the vet, or if somebody's raising sheep or goats or something, they will have a bottle of crystal violet. And if the animal has a wound, they squirt the crystal violet on the wound. I think there's a blue one too that they use, which might be methylene blue. But I've never heard of anybody using it on people anymore. I don't know if they did it back in the day. But if it interrupts cell division, right? If you're exposed to it for a really long time, it's not good. So any of these things, if you're exposed to them in a really high concentration for a long period of time, it's not good for you. Some of them are better than others, but some of them are pretty bad. But it would be like if you're inhaling them a lot every day is what's going to be really, really bad. These are bad. The one thing that we're using right now is we're using quaternary ammonia. What's in the cavi wipes? Do you know what's in the cavi wipes?
No,
No.
I'll look tomorrow.
Yeah, you'll have to look when you go in and see what it says. But the quat, we're using quat, so quaternary ammonia. So they just call it quat. But it is
I just Googled that. It says isopropanol
okay.
and ammonium chloride.
Okay. So that's what, yep. All right.
It kills TB in 3 minutes.
Yeah, so right. So if it kills TB, TB is like, it can hang around for a long time.
That's what it says.
So if it can kill that, that's pretty strong. But it's the combination probably of the alcohol.
Yeah, it's HPV, MERS, HIV.
Yeah.
Everything against, yeah,
So that's good.
nice.
So this is they say this is a cationic detergent. So they're saying this is a surfactant. I didn't know that much about it. At first I was kind of freaking out a little bit. But this is it says, okay, so one problem with quats is that their effectiveness is decreased in the presence of soap, calcium, or magnesium ions, or porous substances such as gauze. And they say it supports the growth of some organisms like Pseudomonas. But that's what they've given us to spray. I don't know. Quats are often now mixed with another agent to overcome some of the problems. They said they used to be in ear piercing kits. It's not supposed to be as bad as bleach. But I don't know. I think most places like hospitals, they rotate through these. So there's not like they're not using the same one all the time. They're doing a rotation. So that's going to help with resistance. Do they rotate in the dentist's office?
No, we usually use cavi wipes or caticide and just,
Yeah, that's
moist
what I thought.
4x4. That seems to be the most effective and
Yeah.
quickest acting, because the biggest problem in dentistry, they want us to clean a room and wait 5 to 10
Right.
minutes.
Right.
And nobody has time for it unless you have a lot of operatories, but
They're so expensive, you can't have that either.
sure. So we are just wiping with a cavi wipes and air dry for a few minutes, but we don't wait 10 minutes. I mean, if there's a cancellation or
Right.
schedule allows, but and you don't always go by schedule because sometimes you run behind.
Right.
So easier said than done.
Yeah. All right. What else, guys?
I don't have anything else at the moment.
Okay.
I don't think I have anything either.
It's 4 o'clock.
I know.
Gosh.
It is 4 o'clock. I didn't realize that.
Yeah.
All this stuff is, to me, it's just cool, but I'm nerdy, right? I think it's
Interesting. Microbiology is very,
it's just really cool, right?
especially now with the virus being around. I think it's just makes you want to learn more.
Yeah, right. And the antivirals, like I put that on didn't I put that on the worksheet for you guys to look up the new one?
Yes.
Yes.
Yeah. It's, I think I saw something today that maybe the FDA did approve it. So you guys might like have 2 different answers because it wasn't approved, but now I think it might be approved.
Oh.
Or they said, they might have said it was okay for something, but I don't know. I wouldn't want to be taking a bunch of a new drug that nobody knows about. I'm just,
Yeah.
it probably won't hurt, but I don't know. I'm too cynical about it all. All right. Oh, here's the thing. Okay, so this is in root canal therapy, that chloramine. So chlorine and ammonia.
Oh, they use a bleach, a diluted bleach.
Really? In that, I didn't know that.
Yeah.
Oh my gosh. I didn't know they bleached it. I just thought they did peroxide.
No, it's half and half bleach and water, distilled water.
Wow. That's crazy. Oh, all right.
I hope you don't need one.
No.
We don't do it at my office, but gosh.
Yeah, right. Well, they send you now, right? They send you to the endodontist.
Endodontist, yeah, it's like a specialist. They have microscope and it's amazing. They did my root canal he didn't even look at my mouth, he looked at a microscope. I was like,
Oh, that's crazy.
yeah, half an
Well,
hour.
and it's so much better though. I had one done, and because they do them all the time, and they can do it quickly, right?
Yes, and I think it's very smart decision to go to the specialist versus having general dentists do it.
Yeah.
They
right.
just not equipped. They can do it, but they can do it the old school way. And if you go
Right.
to the dentist, for them it's like 8 hours a day they do it root canal. So definitely
So
easier,
much better.
and better outcome with more difficult cases. So.
Yeah. I remember the dentist that my mom worked for. I think she's like, he retired and then he went back. He's got to be, I don't know how old he is. He was older than my mom.
I work on Fridays I have 2 jobs, one on Monday to Thursday and then on Fridays.
Uh-huh.
In Silver Spring, that's a killer. 6 a.m. first patient, and 6 to 6. I work 6 to 6 in Silver Spring. It's really hard. But anyway, he's a gum surgeon. He's a gum surgeon. All we do is the gum surgeries all day.
Oh, wow.
75 years old.
75.
Yes, and he just moved to Delaware and commuting from Delaware, getting up at 2 a.m.
Oh my God.
I know. Sometimes I said, Dr. Davis, how are you going to do winter? I mean,
yeah,
he just moved, so I don't know what's going to happen. But 75 years old, but his hands are very steady, very clean. Everything is sterile. He's doing very well for his age. I'm amazed how much. I don't think he works for money anymore.
Yeah, he just does it because he can't. I think that's the way this guy is. I think like a young guy, he sold his practice to a really young guy. And then a young guy came in. I think he's like, wait, you don't know what you're doing. Let me help you out.
That's amazing. This one is the same. Sometimes I just kind of wonder, like in case of emergency, drop on the floor one day. He's like,
right.
I mean, it's 75. He's diabetic and he's doing great, but still, number don't lie. I mean, it's 75, so.
Right.
But we'll see.
That's crazy.
It's interesting to actually see gum surgery, I think.
I think that would be cool to see, actually. And it's amazing now what they do, right?
Yes, it's especially gum graft when they take a gum from palate and the palate is your donor side and you just kind of stitch it up to the front. And it's amazing how within 2-3 weeks, soft tissue can heal and really quickly. And
that's so cool. It's cool, right?
Yes, I love it. Yeah.
Awesome. All right. Okay, you guys have a good.
Thank you.
Yep.
I was gonna ask
Yeah.
about, and it doesn't really pertain to micro, but I was gonna ask if the anatomy chapter 15 has a PowerPoint that goes with it.
Anatomy chapter 15. Oh no, I mean 15 in both books. This is not going to be good. Yeah, 15 in did I not put?
The Learn Smart and everything for Connect is up, but the PowerPoint's not up.
Okay, I'll put it up.
Okay.
Yeah, I have one. I wonder what I did, why I didn't put it there. Yeah, an autonomic nervous system. I definitely have. Yep.
Okay.
And that means there's no videos there then either, right?
No.